Okay, this is the first part of chapter 3, the chapter on biological macromolecules. In this chapter, we're going to be expanding on the information that we covered at the very end of chapter 2 when we talked about organic compounds. So most of the molecules that compose the cells in a living thing are really, really large. And we term these macromolecules. The word root macro here referring to large, and then molecules being these compounds. One of the things that you recall from chapter 2 is that these compounds are known as organic compounds because they also contain lots of covalently bonded carbon. And we call these organic macromolecules or biological macromolecules because of their very, very large size. Some proteins, for example, may contain thousands of covalently bonded carbon atoms. We're going to break the biological macromolecules down into 4 basic categories: carbohydrates, lipids, proteins, and nucleic acids. And you may know some of these by a more common name. For instance, carbohydrates are sometimes called sugars. Things like fats are lumped into the category known as lipids. And proteins and enzymes all fall into this category of proteins. And then nucleic acids are things like DNA and RNA. Now you'll notice that 3 of these 4 categories have an asterisk next to them, and this is because these macromolecules are formed as what we call polymers. So let's talk about what a polymer is. A polymer is a long molecule that consists of many smaller or identical building blocks that are going to be linked together by covalent bonds. The basic unit of these polymers, or these building blocks, are known as monomers. And again, these word roots come from poly, meaning many, and then mono, meaning one. So we group monomers together and we form covalent bonds, sticking them together to make these long polymers. Now, as far as the process for building or synthesizing or breaking down or decomposing these polymers, we're going to use a couple of different types of chemical reactions. So let's look first at how we build these polymers from monomers. We're going to build these polymers from monomers using a reaction called dehydration synthesis. And so basically what's going to happen is we have monomers, and on either end of the monomer we'll find a reactive hydrogen ion or a reactive hydroxide group. And we're going to stick these monomers together using this dehydration synthesis reaction, forming a covalent bond between the adjacent monomers. At the same time, each monomer is going to contribute a part of this water molecule, the hydrogen ion from one and the hydroxide from the other, to make our water molecule. This is why this reaction is known as dehydration synthesis. Now these reactions, the dehydration synthesis reaction, is known as an anabolic reaction. It is a building reaction, and it's going to require the input of energy. We'll see in a later chapter how enzymes are going to be used to help speed up or allow these reactions to progress by lowering the activation energy of the reaction. For right now, you can think of these enzymes as switches that are going to control or regulate when these reactions occur. Now, polymers can be broken down into smaller units by a reverse process, which is a catabolic reaction, and we're going to call this catabolic reaction hydrolysis. This process is going to require the addition of water. In fact, the word hydrolysis refers to using water to split things. And so what we're going to do is we're going to use water to split apart the monomer from the polymer. It is the reverse of dehydration synthesis, and it's also going to require special enzymes in order to activate. So here we can see an example of how this might look. Now again, this is a very simple example. The monomers here are just represented by these individual little spheres. And what you'll notice is that one monomer has a hydroxide on one end and a hydrogen on the other. Okay, so we're going to use a reaction called dehydration synthesis to add this monomer to this growing chain of polymers here. And so the hydroxide from this end and the hydrogen from this end are pulled away, dehydration, to make a water molecule, and that leaves this reactive point here to form this covalent bond that joins this monomer to this growing chain of polymers. What you'll notice also is that no matter how long this chain of polymers is, we have a reactive hydrogen here and a hydroxide on the other side. So we can continue to stick these together like Legos. So this is an anabolic process because it is a building reaction. We're taking two smaller things and building something larger. The reverse of this is a catabolic process where we're taking a larger molecule like this polymer and we are splitting it into two smaller things. We are going to use water to do this, and so of course this reaction is termed hydrolysis. By using the water to split and break apart this covalent bond, the water molecule is broken apart and the hydroxide is added here and the hydrogen is added here. Now remember, these reactions, though the reverse of one another, each require specialized enzymes in order for the reactions to occur. And as we'll see when we start talking about each of the four groups, the processes of building and breaking these polymers down is going to be the same. However, the monomer will vary. For example, if this was a complex carbohydrate, the monomers here would be simple sugars. If this was a nucleic acid that we're going to be building here, the process would involve nucleotides, which are the monomers of nucleic acids. And then finally, if we are going to be building a protein, each one of these monomers would be called an amino acid.